Thank you for kind introduction. My name is Yukihiro Ito. And today I'd like to talk about genetic engineering of rice. The title of my talk is Enhancement of Saccharification Yields from Rice Straw. This shows cycling steps of biorefinery. Biorefinery is a technique to produce fuels or useful chemicals from biomass. And in this case, we use cellulosic biomass. It's plant cell wall. The problem of this step is the difficulty of saccharification from cellulosic biomass because plant cell wall is physically strong and biochemically stable. So for this step, it needs a lot of energy or cost for the saccharification. And if successfully plant biomass is saccarified, then the saccarified sugars, fermentable sugars, can fermented to produce fuels and chemicals. And if polymerization is added, the chemicals, plastics, can be formed. And after the use of fuels chemicals, they are converted to carbon dioxide, which will be absorbed by plants by photosynthesis. So this is recycling steps, and that's sustainable. But the problem is this step, saccharification. So to overcome this problem, a number of studies have been carried out, and we have been studying to modify or to breed material plant to be suitable for biorefinery by enhancing saccharification yields. This is an idea. We assumed if cellulose is overexpressed during plant growth, a partial degradation of cell wall will take place. And such cell wall will be an efficient material for saccharification because by the expression of cellulase, the partial degradation of plant cell wall takes place like this. And such cell wall will be an efficient material because cell wall degrading enzymes will be able to get into the inside of plant cell wall through the cracks generated by the expression of cellulase. But there are two other possibilities. One is that overexpression of cellulase causes overdegradation of cell wall, and in that case, the plant will be dead, so we cannot generate such transgenic plant. And the other possibility is that plant cell wall consists of various components, so expression of cellulase alone is not sufficient to modify plant cell wall. And in that case, we will never observe the enhancement of saccharification yield. So to examine which is the case, first we overexpress the cellulase in rice. And the cellulase is categorized into three types. One is N-glucanase, and the second one is X-glucanase, so which is also called cerebrihydrolase, which produce cerebiose. And the third one is beta-glucosidase, which degrade cerebiose into monosaccharide glucose. So we expressed each of these three cellulases in rice. And this shows the vectors used for rice transformation. And EXG1 is X-glucanase, and ENG1 is N-glucanase, and BEG1 is beta-glucosidase. And all three genes are derived from rice. And for these two genes, ubiquitin promoter is used. And for this gene, BEG1, beta-glucosidase, actin promoter is used. And both ubiquitin promoter and actin promoter are very strong constitutive promoter in rice. And first, we checked transformation efficiency. And in case of EXG1 X-glucanase overexpressing construct, we obtained 13 transformants out of 200 color used for the transformation. So the efficiency was 6.5%, which is comparable to that of empty vector transformed plant. This is a control. And when N-glucanase overexpressing construct was used for the transformation experiment, we obtained no transformant, even 3 times more, 600 color were used for the experiment. So this suggests that overexpression of N-glucanase is toxic to plant cells, probably due to overdegradation of cell wall. And when beta-glucosidase overexpressing construct was used, we obtained 5 transformers, and the frequency was 2.5%, which is more or less comparable to the control plant. But further analysis of the beta-glucosidase overexpressing plant showed no effect of the overexpression, including the saccharification yields. So we focused on our analysis on these plants. And first, we checked the expression of the introduced gene, EXG1. And this is the control plant transformed with an empty vector. And under this condition, no expression was detected in the control plant. But in the overexpressing plants, we observed various levels of the expression of the introduced gene. And the difference of the expression level between the independent transgenic plants is very normal. So this is very normal result. And then we check the cellulose activities in the transgenic plant. This is a control plant, and these are overexpressing plants. And overexpressing plants shows higher cellulose activities than the control plant. So we examine the saccharification yields from these rice straws. And this shows experimental procedures for saccharification analysis. And the first rice straws were dried at 105 degrees. So during this step, endogenous enzymes are inactivated. And then the dried samples were ground into powder. And then the powder was used for the saccharification reaction using commercially available cellulases. And then the concentration of reducing sugars in the reaction mixture was measured. And this shows the result. And the left side is the concentration of glucose in the reaction mixture. And this is reducing sugars, including glucose. And V is the control plant. And these three are transgenic plants overexpressing hexaglucanase. And the plus indicates cellulases, commercially available cellulases added to the reaction mixture. And minus indicates no cellulase was added to the reaction mixture. And as you see here, the hexaglucanase overexpressing plants showed higher saccharification yields compared to the control plant in focusing on glucose or reducing sugars, including glucose. So overexpression of EXG1 hexaglucanase enhanced saccharification yields from rice straws. However, we observed unfavored phenotypes in addition to the enhancement of saccharification yields. First, in leaf, in the control plant, we observed very normal leaf like this. But in the overexpressing plants, we observed separation of leaf like this. And these are scanning electron microscope observation. And in the control plant here, we observed normal surface of the leaf. But in the overexpressing plant, we often observed cracks like this on the surface of the leaf. So from these results, we suggest that the overexpression of hexaglucanase 1 weakened cell wall. In addition to the leaf phenotype, we observed unfavored phenotype in Panicle. And this is a controlled plant, so we observed very normal Panicle. But in the overexpressing plants, the Panicle was very small, like this, and they produce no seed, so they are sterile. So overexpression of EXG1 unfortunately induced sterility. And our purpose is to generate rice plant from which seeds are used as food, and the remaining straws will be used for fuel or chemical production. And this result is good for fuel and chemical production because saccharification yields are enhanced, but bad for food production because they show the sterility. So now we need to overcome this problem, sterility. To overcome this problem, we used SGR promoter instead of the constitutive promoter. This promoter is induced upon senescence because we assumed if cellulase is constitutively overexpressed, including the Panicle development stage, the morphology of Panicle was affected. So if we express cellulase after completion of morphogenesis, we will never observe malformed Panicle. After the completion of the morphogenesis, the cellulase cannot affect the morphology of the Panicle. So we used this SGR promoter, which will be induced by senescence. And first, we check the promoter activity. And so we generate plant with SGR promoter and gas reporter gene. So the gas reporter gene is activated by this promoter upon senescence. We checked this one. And this is the artificial senescence. And the drug treatment induced the senescence. So we treated these leaves with senescence. And this is 0 day after senescence, and 4 days, 7 days, 9 days, and 11 days after senescence. So as you can see here, 9 days and 11 days after senescence, we can detect the reporter gene activity. But before 7 days, 0 day, 4 day, and 7 day, we detected no gas activity. And this is natural senescence. And we used 3 leaves, flag leaf and preceding leaf 1 and 2. And this shows the position of these leaves. And this is here Panicle, and this is flag leaf. This is preceding leaf 1, and then leaf 2. And among these 3 leaves, flag leaf is the youngest, and preceding leaf 2 is the oldest. So this is the most senescent, and this is young, before senescence. And as you can see here, we detected gas activities in the oldest preceding leaf 2 here, but almost no gas activity was detected in the younger flag leaf or preceding leaf 1. So this indicates that this promoter, SGR promoter, can induce gene expression along with senescence. So we make these vectors for rice transformation. This is a constitutive promoter, and this is a SGR promoter which induces the gene expression upon senescence. And this is a control that contains only promoter and no gene. And first, we check the leaf morphology. And this is a controlled plant, and so we can see the very normal leaf. And this is a constitutively overexpressing plant. So again, we detected the separation of leaf here. And these are SGR EXG1 plants. And in these plants, the leaves are very normal, not different from controlled plant. So SGR promoter avoided morphological abnormalities in leaves. And then we check the Panicle morphology. This is a controlled plant, so we observed the normal Panicle. And this is a constitutively overexpressing plant. And so the Panicle is deformed and very small and set no seed. And these are SGR EXG1 plants. And these plants produced very normal panicles, and they set seeds like controlled plant. So SGR promoter avoided sterility and morphological abnormalities in panicle in addition to leaves. So next, we check the cellulose activities before and after senescence. And this is before senescence. And this is a controlled plant, and the cellulose activities in controlled plant was set as 1. So relative activity was shown here. And in these plants, before senescence, almost similar cellulose activity was observed. But after senescence, these 3 plants showed higher cellulose activities than the controlled plant. But this plant showed no enhancement of the cellulose activity. And next, we examine the saccharification yields from rice straws. And this is before senescence. And before senescence, almost similar saccharification yields were observed between the controlled plant and the SGR EXG1 plant. But after senescence, higher saccharification yields were obtained from the transgenic plants compared to the controlled plant, but not this plant. And if we compare the saccharification yields with the cellulose activities, if cellulose activity is higher, the saccharification yields was enhanced. And this plant with no enhancement of cellulose activities showed no enhancement of saccharification yields. So SGR EXG1 plant showed enhanced saccharification yields without affecting panicle development, leaf development, and we observed no any morphological differences between these plants and control plant when the SGR promoter is used. Oh, sorry. And the number 15 and 23 indicates that 15% and 23% of the biomass used for this experiment was really saccharifed to reducing sugars. Okay. Okay. So this is the conclusion. And plant-derived cellulases in combination with an inducible promoter can be used for enhancing saccharification yields of cellulosic biomass. And now we are trying to enhance saccharification yields much more by expressing not a single cellulase, but by the expression of a combination of cellulases, different types of cellulases, to enhance much more yields from rice straws. Okay. Okay. This is the collaborators, and this experiment was carried out in collaboration with Professor Soneki's group in Hirosaki University, and the transgenic plants generated with this experiment will be used with Professor Sonomoto's group in Kyushu University, who study microorganisms. Okay. Thank you for your attention.